Thanks very much for the invitation to come tell you guys a bit about what the IGI is doing, and specifically what my lab and the IGI is working on. Really quickly, I think I know the answer already. Just get a show of hands. How many people have heard of CRISPR or Cas9 in this audience? Yeah. So the thing that might surprise you is, right, you guys are, most of you are scientists, physicians, and so you expected a lot of hands to go up. When I ask the same question to a high school class, it's basically the same response, right? Which is crazy if you think about it. And I think that it's because this sort of thing has exploded out of the lab. People are very excited about it. It's transforming biology, but now popular press is picking up on it. People have heard about it. And the reason that people have started to get so excited about it is in part because it's so easy to use in some ways. At least it's easy to maybe get started with the simple stuff. To give an example of that, I talked about asking high school students about this. We've had high school interns in my lab to volunteer, things like that. They make genome editing reagents now. So in the course of a week, they make a genome editing reagent. They don't do the cell biology to do the editing, but they can make the reagent in a week, and it costs about $50. Okay? That's where we are for genome editing right now. So to put some perspective into this, why is this kind of transformative advance in terms of cost and speed so important? If you think about the revolution in next-gen sequencing, we sort of now have access to this vast library of genetic information, not just for human genomes, but all genomes, right? We can do metagenomics, we can sequence all kinds of funny organisms and try to figure out how things are working. But it's really a speed reading exercise right now. We're reading, we are approaching wisdom, but at the moment we can read very quickly. But if we see typos, if we want to figure out what does this word do, how is this sentence put together, what happens if we change something, we haven't had the tools to do anything about that, at least at the same scale and at the same pace as sequencing. And to me, that's really what the next generation of genome editing means. And that's what makes it very important, is putting find and replace on top of speed reading. So to give you an idea of what we're really interested in the lab, let's say you have some kind of cell, and you have a gene of interest, and you want to know what it does. Well, just down the street at the Allen Institute of Cell Science, they're doing genome editing to endogenously tag basically every protein in the human orpheome, right? So they want to know where does every protein go. And so they're going to put fluorescent tags on in the endogenous context and watch them. And that's a hugely, hugely beneficial way to learn what's going on with all these proteins. It gets around a lot of the problems of overexpression, things like that. In my lab, we're not as much interested in doing this kind of tagging. What we are interested in are very small mutations. So we don't want to necessarily put on a big GFP or a big selection marker, but we want to take, say, patient-observed SNPs, variances between individuals, and introduce them into cells. We also want to do this because, from my point of view, this helps link a lot of what we've been learning biochemically and biophysically, in test tubes, to what happens in cells. And I think this is something that the ISB has really been a front runner in, trying to integrate a very quantitative understanding in terms of more complex networks. And I think that's one of the things that genome editing lets you do. Right? You can make small reductionist changes and introduce them into the complex network and ask, what is the effect of that small reductionist change on a larger phenotype? How do these parts play together? And of course, the other thing that people are very excited about for genome editing is going the other direction. If you have some sort of patient mutation, can you take a causative disease mutation and go back the other direction and cure a genetic disease? And I would just point out that doing this kind of endeavor, tagging a lot of genes, it's not as if it's easy, but it's relatively simple because you have some sort of selection marker. Maybe you have a resistance marker, maybe you have a color you can sort on. However, if you have, let's say you have a non-coding SNP that you want to introduce, and you think this non-coding SNP maybe alters the regulation of a gene, you can't introduce that SNP at the same time you put a marker in there. Because of course the marker is going to, the whole point is to do this in a way that exactly replicates the patient. And so we need to have ways to introduce SNPs or to remove SNPs in a way that does minimal genetic perturbation but is maximally efficient. And that's in large part what I'm going to talk to you about today. So how does gene editing work? Okay? Gene editing all starts with cutting DNA. And so at its heart, a gene editing experiment is really a study in DNA repair. Okay? Part of my background is in DNA replication and repair. And I think it's very, very interesting that as we move into this genome editing era, suddenly everyone in the world is doing DNA repair experiments. The number of DNA repair experiments that are going on these days is now, I mean, it's just orders of magnitude compared bigger than what people had done before and say, an individual lab studies DNA repair. But the problem is it's this big meta-analysis. People are interested in their biology, so they do gene editing to study that biology. And as people do those experiments, we're starting to understand that different sites of the genome are repaired very, very differently. That's complicated for achieving gene editing goals, but it's a very interesting question for basic biology of DNA repair that my lab is interested in. So let's say you make the break. In mammalian cells, people think of this as going down one of two pathways. And that would either be non-homologous end joining, which leads to error-prone insertions or deletions. And I often refer to this as just knocking out a gene. And this is where reagents like Cas9 work extremely well so far, as you'll see in a minute. You can also give a donor template, which can either be duplex DNA or single-stranded DNA. And this might contain your selection marker, your SNP, something like that. And that thing gets copied into the genome, and you can get a precise insertion or a modification or even some sort of very precise deletion. And I tend to refer to this as editing. So when I say gene editing, I often slip into this by accident. When I say gene editing, I'm almost always talking about this. And when I say gene knockout, I'm talking about this. Now, another thing that I would point out is in my lab, we're very interested in, is this really NHEJ and is this really homologous recombination? It's not really clear at this point what's really going on behind these pathways. And I'll tell you a little bit about some of our insights into some of that. We got started off thinking about this sort of historically. Gene editing is not a new thing. So gene editing has been around since the 90s with the introduction of zinc fingers, sort of hit its stride with TALENs and then very rapidly progressed into CRISPRs. And as we started thinking about these reagents, it became sort of apparent to us that previous gene editing reagents were basically symmetric. You would have different recognition motifs on either side. These might be different recognition elements, but at the heart of it, the thing doing the cutting is a heterodimeric or pseudodimeric FOC1 nuclease. You take FOC1, you split it in half, and you make two halves that heterodimerize. But the cleavage is basically symmetric. Cas9, on the other hand, is guided by an RNA, and it has two separate nuclease domains, which are non-symmetric. Okay? They might be in similar locations on the strand, but the active sites are completely asymmetric, and they may be operating independently. And if we look at what Cas9 looks like, this is a 3D model that I had built of Cas9 based on a structure from Martin Ginnick's group, that becomes very apparent. So Cas9 is in white, the guide RNA is in orange, the DNA that it's cleaving is in blue, and you can see that the target DNA strand starts to move down in the Cas9. And if you remove this nuclease domain, you can see the guide RNA wraps all the way around it. Okay? By contrast, this non-target strand is basically exposed. And we started asking the question of basically, what happens after Cas9 makes this cut? Okay? That becomes important to us because we want to understand what's the mechanism by which you get to knockout or to editing. The reason that we're really interested in this is that, like I said before, knockout works pretty well, 20 to 80% depending on what you're trying to work on. We can, in a lot of cases, get this up to 100%. But doing this kind of editing, depending on the cell type and depending on the locus, can be very inefficient. Embryonic stem cells, for example, this is more hovering around 0.01%. And so if you have a selection marker, maybe that's OK. But if you're trying to introduce a SNP and you're trying to then sort through ES cell clones, oh gosh, you're really not going to have a good day when you're doing that. So to try to answer these questions about cellular reductionism, right, introducing specific mutations, how does that affect networks, to try to do this kind of scarless SNP causation of disease alleles, and really to try to enable gene editing, we started asking a lot of questions about how editing is really working. So we start with this super, super simple question. If Cas9 has a target site in the cell, and it binds to that target site, and you get an edit coming out the other side, what happens in that black box in between? Okay? And we're going to start at the very early stages. We're not even going to think about DNA repair yet. For the purposes of this talk, we're just going to say, what is it that Cas9 does? It binds to the site, it cuts the site, and does it let go of both sides? Does it let go of one side? Does it let go of the other side? And depending on what it does, that has a lot of implications for the recognition of that break and processing by host factors. So we want to learn about the mechanisms of repair to make better reagents. So this work was done by a really fantastic postdoc in the lab, Chris Richardson, together with another postdoc, Mark DeWitt, Jordan Ray, who is an incredible undergrad who's on his way to MIT now, and a really great technician, Gemma. So our first surprise was when we looked at the kinetics of binding in vitro. If you take Cas9 or catalytically inactive dCas9 and you bind it to a target piece of DNA in vitro, wild type and dCas9 have essentially the same kinetics, even though one of them is cutting that DNA in half. Okay? Binds, and then it has an incredibly slow off rate. So the off rate here looks basically like a bivalent antibody, even though this is a monomeric protein. So it's almost single turnover at this point. We wanted to make sure this was true, and so we took Cas9, we attach it to a doubly labeled piece of DNA. There's one fluorophore on the plus end, one fluorophore on the minus end. We bind them, we let that go for a little bit, then we add in huge amounts of cold competitor, and we watch to see the dissociation. So this is just an EMSA experiment looking at the dissociation of either the minus strand or the plus strand, and we get the same answer. We get the exact same half-life. It basically doesn't let go. Okay? It's incredibly slow. This is on a non-denaturing gel. If you run this on a denaturing gel, it's very clear that Cas9 has already cut this complex. Okay? So it cuts the DNA, and it seems to be just holding those two strands of DNA together. But what does that look like locally? Because, again, we want to know what does the DNA repair machinery see inside the cell. And the reason we ask this question is this strand is quite exposed. So we started asking, OK, these two ends are globally held together. What's happening on a smaller level to each individual strand? And so in this type of EMSA experiment, what you can see is now we're looking at the shift of either the minus strand or the plus strand. You get a shift, and if you add in duplex competitor, nothing happens to that. But if you add in a competitor that's complementary to the strand that's not bound, you lose the strand, that top strand, the non-target strand, from the complex entirely. It just disappears. And this activity is independent of the so-called protospace or adjacent motif, but it depends entirely on complementarity that's next to it. We do a bunch of controls, we swap labels, this is very surprising, and it all seems to be very robust. And through a lot of various controls and experiments, it turns out what's happening is Cas9 holds on incredibly tightly to the PAM, to both sides of this cut, and it just lets go of this 3' hydroxyl entirely. If we inactivate the enzyme by either making dCas9 or making a nickase such that this strand is intact, instead what we see is bubbling. So Cas9 releases a bubble of DNA, and we can anneal oligos to that as well. We went on, we showed that this happens in cells, so we use end labeling experiments to show that we can indeed label these hydroxyls in cells during genome editing. And the fact that we can see this flap happening in cells and the fact that we can see this kind of annealing happening led us to try the following with HDR experiments. What we do is we take Cas9 recombinant protein, we T7 transcribe a guide RNA, and then we introduce that together with a single-stranded DNA donor. We tend to call this thing the RNP, the Cas9 RNP, ribonucleoprotein complex. We really like this because for us, it's very effective in a whole bunch of different cell lines. As you'll see in a little bit, it's very effective in primary cells, and it even works in, say, mouse zygotes. So we can do genome editing in a matter of weeks to make mouse lines. So we decided to go through, and using this principle of annealing to a released strand, a flapped strand, we specially designed the single-strand DNA donor. Here we have a reporter assay where wild-type cells are up here in this quadrant. When you get a knockout, you drop down to this quadrant. When you get editing, HDR, you would move over to this quadrant. If you just give these cells the RNP, you drop down to here. If you give specially designed annealing oligos that are just long enough to anneal to this region, about 35 bases, and then extend off this direction, so they're very asymmetric, you can get around 60% editing. And if you extend this annealing region back further, so it would have to go into the duplex that's no longer flapped out, everything drops precipitously. If you swap the strandedness, if you take the exact same single-stranded DNA and just swap it so that it would try to anneal to the thing that Cas9 is still bound to, the efficiency drops precipitously. So it seems to have something to do with physical localization to the complex. So this is working extremely well in cell lines. We can introduce SNPs, relatively small regions, couple bases, several amino acids, tags, extremely efficiently, and without having any kind of selection. But does it work in a real application, right? So this is for basic discovery, asking questions about what's happening for basic mechanism, but can we try to do this for something that might be more therapeutically relevant? So now I'm going to switch to telling you about how we've tried to apply this for editing in hematopoietic stem cells to try to develop reagents that can cure sickle cell disease. This is in collaboration with a group at Children's Hospital Oakland Research Institute and UCSF Benioff, David Martin, and Mark Walters. So to give a brief background on sickle cell disease, this is caused by polymerization of a mutant form of hemoglobin, forms long chains, polymers of hemoglobin. This leads to characteristic curvature, sickling of the red blood cells, which clog capillaries. It's a relatively large affected population, about 100,000 people in the US, mostly African Americans because of the genetics of the disease. But in West and Central Africa, 1 in 4 people carry the trait. So you can imagine how many people are born with this. Part of the reason why it's very difficult to say how many people really are born with this disease, though, is that the mortality is estimated at 50 to 80% for those under 5. This was really brought home to me when, during recent round of graduate recruitments, a woman from Nigeria came in for recruitment, and I told her that I was working on sickle cell disease, and she said, "Oh yeah, my brother died of that, and so did my 2 cousins." Right? All of them died in childhood before they were 5. So it's really a terrible disease in Africa. In the US, once you get past the sort of critical years, the primary manifestations are capillary clogging, which leads to really painful crises. You can have acute chest syndrome, which is clogging of the capillaries in the lungs. But also, of course, if you have clogging in the brain, that can lead to stroke. Even in the US, life expectancy is very far down. What I want to point out is that most care for this is only supportive. It's a genetic disease. So you're not actually addressing the root cause. You're just sort of helping people live with the symptoms. This is especially frustrating because we've known the genetics of this disease since 1949. Linus Pauling is actually the first author on the paper to describe the molecular nature of sickle cell disease. So before we even knew the structure of DNA, we knew what causes sickle cell disease. And we knew what led to this. We knew the genetics of it. From a curative point of view, we want to take the SNP, which is a glutamate to a valine SNP, and turn it to either a heterozygote or homozygote wild type. Heterozygotes are carriers, and of course, homozygote wild type have no problems. You can use allogeneic bone marrow transplant. You take a healthy donor, but the problem is it's a relatively restricted patient population. Finding an HLA-matched donor can be difficult, and the people that benefit the most from this tend to, you have to do the transplant relatively early, and that means that since you have to ablate the bone marrow, there are fertility problems later in life. So you sort of give people this choice of, well, you're either going to have sickle cell disease or you're not going to be able to have children. And of course, since this is a bone marrow transplant, there is a small risk of rejection and death. So we really want to develop some sort of therapy that matches the severity of the disease with the safety of the cure. So we want to be able to transplant a patient's own edited tissue. In an autologous transplant, we want to take diseased individuals, do ex vivo gene editing in a very precise way, and then reimplant that tissue back into them. So we want to be having an effect in the red blood cells, but of course, to do that, we need to be working way back here in the hematopoietic stem cells in the bone marrow. The difficulty here is that these are basically unculturable, right? You get stem cells from donors, and they stay stem cells for about a day and a half, and then they start differentiating into different blood components. And people are still trying to figure out why that is, and ways to maintain stem cells, at least hematopoietic stem cells, but so far that has not worked out very well. It also has been seen that these are very difficult to edit. So people have tried for a long time to introduce sickle editing reagents with zinc fingers and talons, and it has not worked out very well. And so we hope that maybe using this new approach, we'd be able to have a little bit more of a boost in that. So what we did was we took the region, we made every single guide RNA in the region that would be amenable to replacing this codon. We again use the RNP, and we use asymmetric HDR donors. So what I'm showing you here is results on either indels, that is, whenever you make a knock-in, you always have some fraction of alleles that also get knocked out instead. What I'm showing you here is starting with an asymmetric donor and moving to an even more asymmetric donor, the fraction of HDR versus indel. And at the best reagent, we can get to editing that's basically indels and HDR are about the same, okay? Right around 35% HDR. We can also do this using very inexpensive unmodified guide RNAs. These are very easy to scale up. Making all of these types of reagents, the whole thing costs maybe $150 to make this, for this small scale type of thing. So this is editing on the genomic level, but what sort of effect does this have on the protein level, on the hemoglobin level, which is what we really care about. And so now we're going to take Sickle HSCs. These are from blood discard from people that have gotten whole blood exchanges, patients. We're going to edit them. We're going to immediately differentiate them on purpose into red blood cells, into erythrocytes. And then we're going to do HPLC to ask about the globin protein. So this is what the HPLC looks like. This is an untreated set of cells, and these are an edited set of cells. And you can see that the sickle protein goes way down, and the wild type hemoglobin goes way up. And if you quantify that on basing it on with increasing asymmetry, again, you can get to right around 40% conversion of the sickle protein into wild type hemoglobin. So we're pretty excited about this. I think there's still a long ways to go, of course, for really curing a genetic disease. Right now we're working on animal models, so stay tuned for some of that stuff. But at least on the genetic level and on actually making a change on the protein level, which is what matters for the disease, we seem to be doing pretty well. So what we do to do all of this editing, like I told you, is we take Cas9 protein, in vitro transcribed guide RNAs. We use these very asymmetric single-strand DNA donors. They're very cheap. They're very fast. We've developed ways to put all of these together in 96 and 384 well format. So at this point, we're doing hundreds to thousands of reagents a day. So we can try a lot of possibilities and do a lot of optimization. That has been pretty important. I didn't show you all of the things that failed in terms of massaging parameters for a lot of the HSCs. We can get pretty good sickle allele correction in HSCs. This can be very effective in primary human cells and most cancer cell lines. Right now, again, we're really interested in mechanisms of this DNA damage repair. It turns out that these single-strand DNA donors do not seem to go through canonical homologous recombination, which is the way people have mostly thought about HDR working for gene editing. So we're trying to figure out exactly what pathway they do use. Because whatever pathway it is, it's extremely effective in a lot of cells. So like I said, we're trying to figure out what mechanisms those are. For the asymmetry point of view, getting into what these mechanisms are, we don't really know if we're repairing some sort of repair intermediate, or are we just increasing the local concentration of donor near the cut. And so we have some experiments to look at that. If anyone is interested in learning much, much more about gene editing, the IGI puts on a workshop every year. This is, the next one is coming up in July. The application deadline is April 10th. Speakers and instructors include myself, Dana Carroll, Jennifer Doudna, Jonathan Weissman, Dan Voitas. So please head to the website and apply. This is actually a lab-based course, and so at the end of the time, students and postdocs will have a chance to actually do gene editing in human cells. So if anyone is interested, I encourage you to apply. With that, I'll thank the people who actually did the work. Again, Chris Richardson did all of the asymmetric donor work. Mark DeWitt was the leader on the hematopoietic stem cell project. And thanks to everyone in the lab and all the really fantastic collaborators that have made this possible. And thank you for your attention.
Can take a few questions. Paul?
Very nice talk. So I'm not sure I followed the rationale for autologous versus for the bone marrow transplant versus doing this from the patient's own stem cells. Because wouldn't you still need to replace all of the stem cells from the patient to get the curative effect?
Yeah, so there's some data that you don't, actually. So there are people who have mixed chimerism. And so that is where you do an autologous bone marrow transplant. And they show benefit even when they have only 2% of donor cells. Okay? People have shown basically complete cure with as little as 8%. So the idea is that if you're doing autologous, you want to clear everything out because you need to clear a niche for the new cells, right? But if you're basically taking a certain portion of your own cells and just putting those in, maybe you don't need to clear out the entire niche because you're not going to have competition between the populations.
So having only 2 to 8% normal red blood cells is enough to avoid the...
So this is because, so one of the major manifestations of sickle cell is anemia. So a normal red blood cell lasts in the blood much, much, much longer, 5 to 10 times longer than a sickle red blood cell. And so even though you're still continuing to make sickle red blood cells, they get made and they go away. And your body has feedback mechanisms for hematopoiesis. And so you basically boost hematopoiesis, and the population of normal versus sickle starts to dominate normal, even with relatively low fractions of HSCs. Yeah. The exact numbers, 2 to 8, that's sort of debatable in the clinical field, but people have seen clinical benefit, like I said, with 2, and by 8 to 10, people seem basically fine.
As you pointed out, stem cells are hard to culture. Stem cells are hard to culture. Could you visualize moving from ex vivo treatments to in vivo treatments, bone injection?
Yeah, absolutely. I think that's a great question, and that's something that we're trying to do right now. Like I pointed out, most of the people that have this disease are in Africa, right? Central Africa. And they're not going to be getting bone marrow transplants, right? That's not tenable, especially for 15 million people. So we're very interested in developing some sort of delivery mechanism that can home to the bone marrow rather than requiring ex vivo treatment. Absolutely. Yeah.
Sorry, but what was the hemoglobin F peak? Is that just
Oh, yeah. Wow, you have...
experiment?
No, you have a really good eye. So it turns out that, okay, so the background for this is hemoglobin F is fetal hemoglobin. And it turns out fetal hemoglobin is also protective against sickle cell disease. So normally, when you're a child, when you're just born, you switch from fetal hemoglobin to adult hemoglobin. And fetal hemoglobin expression protects against sickle cell. And what we found from some of these editing reagents is that if you induce partial thalassemia by cutting inside beta globin, you actually induce substantial fetal hemoglobin. And we're still trying to figure out the feedback of that. There are several patients who have thalassemia that induce, I mean, this is one of the hallmarks of thalassemia, is induction of fetal hemoglobin. It's, from a therapeutic point of view, it's dangerous, right? Because you're going to take someone with sickle and say, well, we're going to give you thalassemia and hope that you express fetal. So right now we're still in a very let's understand mechanisms of that before we think about going.
Well, years ago, Tim Lay and Art Nienhaus showed they've used hydroxyurea as a way
Yep.
of stimulating hemoglobin F, and et cetera. I mean, it's sort of an interesting observation. I wouldn't have expected to see it in this
Yeah.
context.
We expected to see a little bit of induction, not nearly the levels that we saw. Yeah, I mean,
It's a lot.
yeah, exactly. So there are these other strategies to do nothing but increase fetal hemoglobin. From what we can see, the amount of fetal hemoglobin induction we get with certain gene editing reagents is equivalent to those other strategies. But there's something really interesting going on. So this is sort of all unpublished fun data, and so take it with a grain of salt. Certain gene editing reagents do this and others don't. And it seems to have to do with the nature of what happens to the message. So when we do RNA-seq on the erythrocytes, what we see is that some gene editing reagents induce some spectrum of indels that lead to NMD, nonsense-mediated decay, of the beta globin message, whereas others, the message sticks around. In the case where we get NMD, so that the beta globin message goes away, we get huge spikes in fetal hemoglobin.
And we can talk about this later, but somehow the inside of the cell has to know
Yeah.
and upregulate transcription in a relatively specific way in the globin locus, which is...
Yep.
is-
Absolutely.
Certain people here know a lot about the globin locus.
I have a couple of questions. So the Cas9 protein needs to be present in the cell to affect the NICs and mediate the translocation and so forth. So do you put the protein in, or do you have heterologous expression that you introduce?
We electroporate the protein in. And the reason we do that is we're sort of looking down the road at where we eventually want to be therapeutically. There are a lot of different delivery vehicles for Cas9 and for gene editing. AAV is one of them. One thing that we have seen, though, is this long-term expression may be one of the things that underlies some of the off-targets people have reported from Cas9. Okay? If you think about our genes, there are only ever 2 targets you want to hit, right? And anything more than 2 is only going to be bad news, right? And so you really don't want something that introduces an editing reagent and then sticks around for days and days and days when the edits happen within the first 2 hours. So from what we can tell, when we introduce these protein reagents, they're around for about 12 hours, and then they're extremely rapidly cleared by the cell. So it's more like a short, sharp shock of editing that seems to be associated in most of the contexts that we've looked, that we haven't been very exhaustive, with reduced off targets. And it also pairs well. We've started to make some of these higher fidelity Cas9 variants, and those seem to synergize really well with that. So that's sort of where we're going. That, of course, raises other delivery issues of now, instead of AAV, you have to get a protein in, and that's difficult. But one of the nice things about being at Berkeley is there are a lot of really talented bioengineers who are doing nothing but nanoparticle delivery systems and things like that. So we have some really nice collaborations on that front.
Well, thank you.
Thank you.